What is up, everybody? Welcome back to Wildlife Cake and Cocktails, our first Quarantinis show, coming to you from the podcast basement, all quarantined up. It has been a bit of a road getting here, to be honest. We've had to, like everybody else, adapt a little bit. We've got all kinds of new equipment and gadgets running, and my producer is not even here. He's helping me out remotely. Christian is doing an amazing job helping me set this up. So thank you again to Christian Summers, our intrepid producer here at Wildlife Cake and Cocktails. And we are back in a slightly weird format for what we are calling the first of the Quarantinis. So I have a lovely gin martini here. Ooh, and it is on fire. I also have a coffee chocolate cupcake with a coffee buttercream right here. Look at this thing. Wow. The joys of working from home right there. Woo. So look, we have this new setup. We've got plenty of new shows coming at you. You can stay up to date. Follow us on Facebook, Instagram, and Twitter. The podcast available at Apple Podcasts, Spotify, and more. And our guest today is going to be Mr. Scott Eipper from Nature for You. He's a herpetologist, naturalist, author, and has written multiple books on venomous creatures, snakes, frogs, and more. Along with his partner, Ty, he operates Nature for You from here in Brisbane. They're a wildlife education and consulting company operating throughout Queensland and throughout Australia. You can find them on Facebook at Wildlife Demonstrations or at wildlifedemonstrations.com. We're going to get him in now. Bring him in. If technology is willing, let's see.
Hello.
Hello.
How's it going?
Good, Scott. Hi, thanks for joining us, man. How are you?
Good, mate.
Excellent. Great to finally have you on. I know we've had some teething problems, but I think everybody's learning to pivot and adapt a little bit these days. How are you doing with all of this mayhem?
Good, mate. I suppose the one thing with this self-isolating and stuff like that, I mean, we're both essential workers, so we're still out and about a little bit, but we're minimising the amount of interaction that we need to do. That being said, though, we're trying to minimise our interactions with our venomous snakes as well at home. So obviously doing anything that needs to be done, routine husbandry and all that sort of stuff still needs to happen, but we're avoiding taking photos and the non-essential items, I suppose. It's a good thing that's coming down into winter. So coming down into winter means that we're less feeding and all that sort of stuff as well. So from our points, it's something to keep in mind, and certainly depending on where we're at in a few months' time will depend on what we're doing with regards to breeding this year as well, because essentially you want to minimise non-essential interaction with these things. Hospital waiting rooms and hospital resources are going to be few and far between if the curve has a secondary peak, I suppose. Obviously it's flattened out now a fair bit, but if it does have a secondary peak, we don't want to be burdening emergency wards or anything like that with snakebite wherever we donate.
Well, yeah, that's something that you and I discussed, I think, already previously, right? Like the issues with respiratory distress from venom-induced consumption, coagulopathy, and all of the other things. Nobody wants to be on a respirator at the best of times, for snakebite in particular. But yeah, this would really be a terrible time to be admitted to hospital for a snakebite for something that was. I mean, most snake bites are generally avoidable, right? So there's no reason to not have that little bit of extra caution, particularly with everything that's going on, right?
Well, it's all about risk minimisation. And at the end of the day, you're young, I'm reasonably young. The chances of us getting sick from COVID-19 are probably pretty slim. But however, we could be carrying it. We pass it on to somebody else that isn't in the boat that we're in, and then suddenly they have some really serious symptoms. We all know people that have depressed immune systems for whatever reason. It could be medical reasons, it could be age, it could be all sorts of things that hit at it, and we don't need to be carrying that stuff around where we don't need to.
Yeah, no, absolutely. And as you mentioned, both of us are still doing the snake removal thing where needed, as essential workers, as we've been classed. But having to put in a lot of extra PPE, a lot of extra precautions, asking our clients a lot of questions, slightly invasive questions about their current medical history and their travel history over the last 14 days and things like that. I guess I know that you guys have also been affected by this whole thing. You guys had some pretty big travel plans with yourself and your partner Ty, your co-author as well, and your amazing partner there at Nature for You, who does a lot of the husbandry and feeding work with you there as well. I hear you guys had a bit of a book tour planned there.
We're meant to be touching down in LA as of about 20 minutes ago.
Oh wow! I had no idea that was
like literally today. We got an email from the flight people saying make sure you don't miss your flight. That was a day and a half ago.
So I guess we're missing.
And I was looking on Stalkbook earlier today, and one of my mates over there had been out chasing red diamondback rattlesnakes, and I'm sort of sitting there thinking to myself, that could have been us.
Wow.
We're actually going to be staying with him as well, so it's pretty shitty, but what do you do?
And you guys were going over there for a reptile conference because of what was it? I believe it was this one.
So we were doing I was actually doing a talk on varanids in captivity, and also a talk on blue tongues in captivity, and it was to do with how we have changed our husbandry style due to seeing these animals in the wild. So I've seen all the blue tongues in the wild, bar a couple of the Indonesian ones and Adelaide blueies, and I've seen about 18 species of monitor in the wild as well. So because I've seen a swag of sort of both of them, and I mean, you've seen our pits at home and stuff like that, we have changed a lot of our systems around to suit natural behaviours and stuff that was seen from the wild. So the talks.
What are some of the examples of that? I know that even with your terrestrial elapids, I see you guys putting in a lot of like arboreal hides, or like, well, not arboreal, but elevated and just, I guess, more enriched environments for even something that somebody would consider maybe a terrestrial elapid, like eastern brown, which is like we also, brown snakes don't particularly climb, but then you occasionally do see them follow a rodent up a tree, or you find them under some bark on like an old uke or something like that. Like they're perfectly capable climbers, but I guess
so without
what other than like I guess a little bit of extra height is something that you've added for the monitors.
So with all husbandry, with all our reptile husbandry, we've taken the idea that we can always do better. Regardless of whatever you've done, you can always improve on it. Now, it depends. Reptiles are a lot more switched on than what people realise, and they can do a lot more, and they pick up on a lot more than what people tend to realise as well. They're not these brain-dead animals that are just sitting there in an enclosure wasting away their time. So in the 70s and 80s, there was a big push for zoos and stuff like that to change the management of their captive mammals and include enrichment as part of the essential husbandry techniques, I suppose. So it's not only is it food, water, cleanliness, appropriate temperature, appropriate size enclosures, but it's also important to enrich that enclosure and give the animals something to do. And they've shown that there's elevated instances of brain activity, which is sort of a suggestion that the mental health of the animal is probably better. being enriched. So there's a couple of things that we do. We move shed skins around in the enclosures. So what we'll do is we'll take a shed skin from, say, a taipan and throw it in with a mulga snake. And the mulga snake gets all excited because it's now got this fresh scent of something that it's not used to smelling in its enclosure. We'll use different hide boxes and move the hide boxes amongst the different enclosures so there's different smells.
That's an amazing bit of aromatherapy.
Yeah, it's a very different piece of aromatherapy. So, by putting a mulga snake skin into a cage of a brown snake, you've now added a predator smell into that enclosure, which causes the brown snake to behave a little bit differently. Don't leave it in there the whole time, but then we take it back out again. When we're introducing food into the enclosure, we drag the food around inside the enclosure before leaving it in a spot. The reason we do that is we're not trying to actually initiate an excited food response, but we're trying to get that animal, as it's moving around in the enclosure, to hit that scent trail and then follow that scent trail to the prey item. So
like natural tracking response.
Yeah, yes. And then we'll also try things like leaf litter every now and then. The arboreal hides is something that we put into a lot of our enclosures to provide them something different. So not only does it mean that they're just moving around on the ground, but they've got to pull themselves up and sit into something a little bit different. It's quite tight for most of the animals that we have them in. And they can actually get on top of it as well. And so you got like a crevice on the top, and then you've got the tube itself. And so you've got a little bit of difference there for the animals. Initially, we found it was really good for the pythons, but we ended up putting it in on the elapid enclosures as well. And, it's quite unusual, but you do see red bellies sitting up, curled up in them. You see taipans curled up in them, brown snakes curled up in them. They use them. And so the whole idea of that is that if that's an improvement, then hey, why not? With regards to the monitors, we put, again, elevated hides and stuff like that inside the enclosures. Outside, we've done that as well. We've got enclosures that have got hide boxes below the ground level. So that way they're sort of burrowing down and into things. We will put snake sheds into the goanna enclosures. We'll put we'll get blue tongue feces and rub that through the ground and stuff like that. Really important with things like parentes, because parentes' main diet in the wild is blue tongues. So by putting in some blue tongue scents into the parente enclosures, you'll get lots of digging activity and you'll get lots of different behaviours. With Easter, I know it seemed like a bit of a joke, but we got some eggs, and the eggs we actually dyed with some food dye, put into the enclosure. Completely different thing for a round, rolling around bird egg for the parente to sort of tackle. And so it's trying to pick them up and it's slipping in its jaws and all the rest of it. But that presented a different challenge for that lizard. Obviously, they get eggs in the wild, so that's something different. The other thing we try to do as well, you guys ever seen the Kongs that you use for dogs?
Yeah, the chewable toy that you can insert treats or peanut butter or stuff in, and they're used for fairly highly powerful jawed dogs in general. It's a fairly tough material.
So we'll get dead pinky mice, and we'll stick a couple of dead pinky mice inside the Kong, and we'll put that in with things like our yellow spotted monitor or something like that, and the Kong itself is bigger than the yellow spotted monitor's head, so there's no way it could get that out of there. But it spends a good couple of hours trying to figure out a way of getting those pink mice out of that Kong. So again, it's something different. It's a different challenge for the animal to sort of to crack onto. So if we can do something that makes it better, then why not?
Super interesting, man. And I guess a bit of a shame that you guys couldn't go and present this, obviously, overseas. What was the conference that you and Ty were going to?
So it's a herpetologist conference. So basically, it's to do with captive management of reptiles. Essentially a conference that's put together where people come along to it, and then they sort of see some of the leaders in the industry from around the world presenting on reptile husbandry and amphibian husbandry. And the audience itself is made up by the speakers, but also by paid participants, by paid people that come along to it to want to see what's going on the cutting edge, I suppose.
Very cool. Very cool. I hope it gets rescheduled. Is there plans for you guys to go back over there at some point?
Yeah. So at the moment, everything's just been put on hold. So hopefully we'll know where it's at and get our flights booked again and go over. I want it to be around this time of year though, because the herping's much better this time of year.
Yeah, a little bit warmer, right? Was there a few target species that you guys were hoping to find while you were over there?
Yeah, anything that shakes its tail in defense.
Yeah.
No, we were going to be chasing,
addicts, right?
Yeah, so we were chasing rattlesnakes and Gila monsters, and while we're over there too, we were going to catch up with some captive collections over there as well, and fuck around with cobras and bushmasters and mambas and all sorts of stuff. So I'd already spoken to to one of the anti-venom banks and stuff like that as well, and tried organising a membership and stuff like that, so that if something happened when I was fucking around with something, then at least we'd be okay. So yeah, a little bit disappointing, but hey, it is what it is. We're both busy working away on another project at the moment, so until such time, we're still very busy.
So more publications from you guys on the way?
Yeah, there's another one happening right now. So
awesome. So at least keeping you guys both busy in the quarantine a little bit.
Yeah, definitely.
All right, cool. Well, look, we actually have quite a number of papers to get through, so we should probably jump into this. And the first one's kind of interesting, right? Because with all the issues that we're currently experiencing with COVID-19, there's some discussions about like the safety of wet markets. And I know that at one point there was and again, this is proven to be incorrect, but people thought that the coronavirus may have jumped from bats to snakes to people. But that was obviously proven incorrect. It was looking at the percentage similarity of the ACE spike protein, not the whole genome. And even then, there was a very low percentage similarity compared to what you actually need to find the true host virus reservoir. So definitely not from snakes, but there might be other potential issues with eating snakes. Not necessarily a great idea, it turns out. And we will just jump into. Let me see if I can pull this up here. Bear with us, guys. We're muddling our way through here, but we're going to pull up our first paper right here. So this is from Letoof et al. From 2020. Science of the Total Environment. Toxic time bombs. Frequent detection of anticoagulant rodenticides in urban reptiles at multiple trophic levels. Interesting paper from the folks down there at the Curtin University in Perth. They do some amazing reptile work over there in the southwest corner. So straight onto the abstract. Anticoagulant rodenticides are regularly used around the world to control pest mammals. Second generation anticoagulant rodenticides are highly persistent in biological tissue and have a high potential for bioaccumulation and biomagnification. Consequently, exposure and poisoning of non-target organisms has been frequently documented, especially in countries with unregulated AR sales and usage. Most of this research has focused on rodent predators, usually raptors and predatory mammals, although exposure has been documented in invertebrates and insectivorous fauna. Few studies have explored non-target exposure in reptiles, despite species sharing similar trophic positions and dietary preferences to other exposed fauna. We test 3 abundant urban reptiles in Perth, WA, that differ in diet and trophic tiers for multiple AR exposure. The Juga, Pseudonar finis, which is a rodent predator. The bobtail, Tiliqua rugosa, which is an omnivore. The tiger snake, Notechus scutatus occidentalis, which is a frog predator. We found frequent exposure in all three species, 91% in dugats, 60% in bobtails, 45% in tiger snakes. Mean combined liver concentrations of ARs of exposed individuals were 0.178 milligrams per kilogram in dugats, 0.40 in bobtails, 0.009 in tiger snakes. High exposure frequency and liver concentrations were expected for the dugite, obviously being a rodent predator, right?
Yeah.
Exposure in the other species is more surprising and implies widespread AR contamination of the food web. We discussed the likelihood of global AR exposure of urban reptiles, highlight the potential for reptiles to be important vectors of AR in the food web, and highlight implications for humans consuming wild reptiles. Scott, first thoughts, mate?
Very interesting and somewhat scary paper in a lot of ways. I suppose the concern is that how far those ARs have sort of travelled through the food web, in that, you know, species such as tiger snakes that don't eat a hell of a lot of rodents are having these rodenticides sitting in their bodies. It's interesting how the paper postulates that it could be from essentially that it might be getting through frogs and the frogs are picking it up through their skin, and then the snakes eat the frogs, and then the frogs are then they're getting those ARs. Then it's certainly an interesting thing. The concern is how long in reptiles that these ARs can sort of persist in comparison to mammals, where they tend to move out fairly quickly. In reptiles, they seem to stay a very long time.
Yeah, and that was quite interesting. I think they had the reference to lava lizards on the Galapagos Islands, where they baited for rodents on the islands, and a number of native birds got poisoned, but there was also a number of lizards that showed toxicity for, was it like over 400 days?
At 800 days post-baiting, liver concentrations between 0.001 and 0.2 milligrams per
800 days.
At 800 days post-baiting.
Yeah, and those liver concentrations were high enough that they would potentially be of risk to something eating.
Yeah, so I think the 0.1 of a milligram per kilo was shown to be toxic in mammals and birds, depending on the taxa. And so these lizards, 800 days post-baiting, can have up to 0.2 milligrams per kilo sitting in their livers. So that's some pretty crazy sort of toxicity. 800 days post-baiting, they're effectively become poisonous, I suppose.
Yeah. And it is interesting, right? Like, so 91% of dugat's, which are largely a rodent predator, I guess in urban areas, you would expect there to be some occasional accidental filtering in of rodenticides, but 91% is just so high. And then you've got the tiger snakes, which was still at 45%, which I guess the question is that happening from the diet of tiger snakes, which we think is generally more amphibian based, but like there's got to be the occasional rodent thrown in there, right? Like we all know.
Yeah, I mean, they'll eat
slightly gutsy eaters. they'll eat a lot of stuff.
A lot of tigers do eat a lot of frogs. The frogs sort of are their main diet, but they are opportunistic. I suppose too, it depends on the location. You know, if you go on some of those islands, tiger snakes are mainly bird feeders, so you know they are very opportunistic. The one thing that I did find interesting is that they stated that predation on larger snakes is likely to be rare, especially in urban environments. The one thing that does predate on large snakes is larger snakes. So certainly dugites and mulga snakes and stuff like that are more than happy to eat smaller dugites and things like tiger snakes when they come into contact with them. So potentially that's another route of ARs getting into the dugites. Could have been from tiger snakes and bobtails they actually consume.
Yeah, right. So from snakes eating snakes and those snakes having eaten the ARs themselves.
Yep.
So the other thing about their slow passage, obviously this makes a fair bit of sense in reptiles. They have a little bit of a slower metabolism and a little bit of a slower rate of passing waste. They don't, these anticoagulant rodenticides generally excreted out of the body through the feces. So
Yep.
if you're not dropping a lot of fecal matter, you're going to retain those rodenticides a lot longer. So reptiles actually make a fairly good way for those kind of rodenticides to kind of spread even further, right? Like you got to think of maybe some of the native predatory birds, tawny frogmouths, owls, and things like that, which feed on our native snake fauna, are now going to be picking up potentially dangerous amounts of anticoagulant rodenticides.
So I suppose the one thing that is I suppose is something that I don't really know too much about is that where are these rodenticides being concentrated in the body? Is it only in the liver, or is it going throughout the tissue?
I don't know. I think you get the higher concentrations in the liver. So, but I mean that makes sense for something that's an organ that's trying to filter out all the toxins.
Trying to filter it out. So
yeah.
So I suppose where I'm going with that is that
or whether it absorbs into fatty tissues or something like that.
And that's the thing. So if it's sitting in the fatty tissues, which is distributed basically essentially throughout the body, then the concern potentially is that if a dugite or a tiger snake gets hit by a car, and you've got not only do you have the larger carrion feeders, but then you've also got the insects and stuff like that are feeding on that carrion. Those insects get eaten by other lizards, smaller birds, insectivorous birds. It really shows how it can be distributed throughout the food web. In addition to that, there could be, it may not necessarily kill them, but it may make them feel sick. If they feel sick, then are they able to get away from a car in time? Are they able to get away from a cat? Are they able to get away from a fox? So you could get increased predation through lethargy, through sickness, I suppose, due to this as well.
I think that was one of the things I considered as well, is like, are they getting a higher rate of sampling of snakes that have higher rodenticide rates in them just because those animals are more likely to be picked up in their study because they're less mobile, more likely to get picked up by wildlife carers, more likely to be injured. So that in itself might be impacting the sampling of the study, right?
Well, yes and no. I mean, I would think that the amount of snakes that Damien in particular, the amount of tiger snakes that Damien has caught through his work on tiger snakes in the Perth region, you would think if anyone was going to be able to gauge if the animals were lethargic and that the animals he collected were basically because they were a bit dopey from this higher AR concentration, then he would be the one to be able to know that. But
well, I mean, there's even the idea of maybe like behavioral fever, right? Like some of them are more likely to be out and exposed to try to bring their temperature up to try to deal with whatever toxicity is going on or something like that.
Well, I mean, you know that reptiles' immune systems is more functional at the closer they are to their
ideal temperature.
And so, if the animal's a bit cold, it's going to be a bit slow. But if it's in trying to bask earlier or bask for longer to maintain that higher temperature all the way through, then potentially that could be a factor.
Yeah, interesting. Let me see if I can pull this up again, and we'll just jump down to the conclusion briefly. Great study, and very, very interesting. You can see the kind of concentrations here in the graph. You can see some of those black concentrations, some of those black bars, getting some really high concentrations in kind of some of those urbanized areas. there you can kind of see the overall rates of the 3 different species that were studied. So tiger snakes, not so much. Dugat's definitely leading the charge right there. All right. So on to our conclusion. The study offers convincing evidence that urban reptiles offer different different trophic tiers and diet are exposed to residentially used ARs, and suggests the surrounding food web is more contaminated than previously assumed. We predict a similar AR exposure in reptiles of the same ecological niche in cities where purchase of ARs are unrestricted and retail available. Based on their probable resistance to toxicity, low elimination rates, and multiple trophic positions, we consider reptiles in proximity to AR sources, i.e. urbanization, to be good indicators of food web contamination. Our data highlight a novel threat faced by reptile predators and humans consuming wild reptiles captured near human habitation, particularly if liver or fat tissues are consumed. And obviously some further suggestions for some further work. Interesting paper. Final thoughts?
Yeah, I think that their conclusion in regards to testing of ARs in livers from reptiles sold in meat markets in Southeast Asia and also places like South America and Africa would definitely give people a better understanding of what they would also be potentially ingesting as well. And obviously there's going to be health risks and health problems associated with eating rodenticides in humans as well, no doubt.
Yeah, and I guess it also shows that if you maybe look at cities with differential rates of anticoagulant rodenticide production or use, you can probably look at the snake fauna in those areas and see if there's a correlation to see how far into the environment those rodenticides are spreading through the reptiles that are eating the rodents that are being targeted.
Well, not only that, but also too where you've got where you do have seemingly a slow decline in predators in a location. It might be a way of mammalian predators, I should say, that do eat snakes and things like that. It might be a way of going to see if that is potentially one of the causes of that decline in those sort of higher predators.
Yeah, for sure. Interesting stuff. Great paper. But I think we do have to move on to our next one, which is going to be a good one from Nankivell et al. This is in the journal Zootaxa. A new species of turtle-headed sea snake, Amidocephalus. Scott made sure I get that one right. Amidocephalus elapidae. He was on my case about that one. Endemic to WA. This is from Nankivell et al. We describe a new species of turtle-headed sea snake, Amidocephalus oriaris, species nov from Western Australia in the family Elapidae, from the Coral Coast, Pilbara, and Kimberley regions. Phylogenetic analyses of mitochondrial markers place the new species as the sister lineage to the 2 currently recognized species of Amidocephalus, E. annulatus from the Timor Reefs and Coral Sea, and E. ijime from the Ryukyu Islands of Japan. Analysis of nuclear SMP data from the new species and E. annulatus from Australia and New Caledonia provides additional independent evidence of their evolutionary distinctiveness. The new taxon is usually morphologically diagnosable from its congeners by a combination of scalation and color pattern characters, and appears to reach greater total lengths over 1 meter in the new species versus typically around 80 centimeters in annulatus and ijime. The new species is known largely from soft-bottom trawl grounds, unlike E. annulatus and ijime, which usually inhabit coral reefs. The discovery of this new species brings the number of sea snakes endemic to WA to 6. So that's 6 sea snakes at WA. Let's have a look at this guy, because the Amidocephalus, cool genus of snakes, right?
Yeah, very interesting sea snakes. They're quite derived in that they've evolved to specialize eating fish eggs, and so one of the results in that is that they their venom apparatus and their venom has become highly derived, and they haven't put as much effort into fangs and venom glands, I suppose. So they've become much less toxic than their close relatives.
It's an interesting evolutionary shift, right? Because we as snake people, we know of sea snakes as being highly toxic because their prey just has so many opportunities to escape, basically in 3 dimensions. Whereas on the surface of the ground, there's not quite as many options. Whereas a fish can swim up, down, left, right, into the coral reef, under the sand, and it's quite easy to lose any prey that you've invested venom into capturing. So I think that's one of the reasons why we think there's a selective pressure for sea snakes to kind of evolve all of this amazing weaponry for hunting. And then you have the Emetocephalus, which eat caviar, fish eggs, generally scraping them off the coral reef. You can kind of have if you can see the lip here on the go back to this beautiful picture. You can see this this fused lip, which in most snakes is multiple scales. In this guy, it's a big fused scraper, and they have this weird downturned rostral scale at the front as well. I guess
that rostral scale in the front there is only in the males that they have that.
Oh, right. So that might be
there's a horn projection on the males,
right?
And that's used in reproduction.
In reproduction.
Yeah.
Wow. So well, I mean, you can definitely see all the changes that's going on with the head of these animals. The parietal scales at the back behind the eye. There's no big bulging venom glands or anything like that, and they're very interesting. As soon as you stop chasing fish around and just eating fish eggs, you lose the need for all that venom, and it just it eventually mutates away. Literally, like there's I believe there was a study that showed that some of the like 3-finger neurotoxin genes that are really active in some of our more potently venomous snakes have a stop codon really early in the gene. So instead of building the gene, you would just get to a little signal that says stop, and all you're going to build is a little bit of useless amino acid. So that thing is probably, I imagine the transcription factors for it now are even being kind of down regulated and switched off. So, there's.
I think the other thing that's really sort of to be noted as well when we're talking about the scalation morphology is the presence of these apical pits that are basically all over the head of the snake.
Yeah, and we can kind of see those here. I'm going to zoom in a bit more, but I believe you have a better photo as well.
I was going to say, do we want to try and jump? I'll try and jump screens and see what happens.
Yeah, let's just have a quick look here. So you can see some of the spots here, but let's have a look at your Emetocephalus ariaris.
So if you're sharing screen, it won't let me share.
Out here. Bam. There we go.
Okay.
Let's see.
Okay, so let's try this again. We're getting very technical here, mate.
Yeah. All right.
So
yeah, there we
hopefully go. We can see that.
Beautiful thing. Wow.
So if
a much darker individual as well. It looks like its eyes maybe clouded over, like it's ready to shed, or is this a preserved specimen?
Well, that's a dugite. What happened? I don't know what happened then. It just bounced.
Where'd the snake go? Oh no. Well, that's a nice dugite.
That's a tiger snake. Don't need to look at tiger snakes. There we go. Right. We're looking at the animal now.
There we go. There it is. Yeah, look at that. That's beautiful.
Let's zoom in on that, because at the moment, so if people can see the cursor, all these tiny little specks here are apical pits. It's been postulated that these apical pits might actually be sensitive to pressure waves and things like that, and so that might actually help the sea snakes be aware of predators and things like that are approaching them, particularly where water is a bit murky. In the case of Emetocephalus, they tend to live on coral reefs, and in the case of that new taxa, they live on seagrass beds, and so the water tends to be fairly clear. This particular animal that we're looking at here is a Emetocephalus annulatus from probably New Caledonia. You'll notice that this has got a separated a separation between the first supralabial scale and the preocular. That separation is not usually present in Oriaris. So in Oriaris, when we go back to your screen in a second, you'll notice that separation is not there. And so that's essentially the morphological difference, besides obviously the distributional differences and genetic differences between the two taxons.
Right. So we'll have a look at some of those genetic differences in the paper in a moment. But yeah, keep that scalation in mind when we go back. We'll actually have a look at the diagram. Here we go. So you've got the annulatus up the top that we were just looking at on Scott's screen. And it's got this kind of configuration right here up the top. And the new species looks more like this, where the labial scale and the ocular scale touch, and the nasal scale does not touch this larger fused set of labial scales. Interesting. This is, we're going to have a quick look here. They did some pretty fantastic phylogenies here as well. You can see the Ijime right here in the ND4 maximum credibility tree. You've got Ijime, which is the Japanese species, and this new species all the way up over here. Quite separate from annulatus over here in the Coral Sea and Timor Sea, which you can see group into two different species. Now, I believe this ND4 was a little bit incomplete. And then this is the maximum likelihood tree from nuclear. Is this from nuclear data that they did? Either way, you can see that there's a new species down here. Again, fairly separate from annulatus over here as well, right? And fairly well supported in this phylogeny, 100%. So yeah, they did a fantastic. I just generally love these very simple scalation drawings of snake heads, and they're just always good to look at. All right, just here we go. Just having a look here, we can see our new species over here, and then over here, and then annulatus and Timor Sea and Ijime down here on the bottom right.
So if you're looking at there at that that animal from in C, that's a male. So you can see what I was talking about with that rostral horn projection there on the end of the snout there.
Right. So that's only in the males, and the females is just kind of that little downturned rostral scale.
Yeah, correct.
Wonderful.
So the males are always horny.
Isn't there like
I'm a dad, mate. You got to have dad jokes every now and
then. there's too many horned vipers and like. All right, cool. Well, look, let's move on to our next paper here. Either way, cool to know. 6 new species of.
Hang on. Before you do, there's something that I want to touch on just with this. The nomenclature is interesting on this one as well.
Oh, of course.
There's a paper that's sitting before the ICZN, ICZN, which is the International Commission for Zoological Nomenclature, by Kaiser et al. With regards to some cobras in Africa. The animals were named by through conventional methods, I suppose, if you will, and then there's also been a private person that's gone ahead and published some names. A lot of places are choosing not to recognise those names for whatever reason it is, whether it be politics or whatever. It's not for us to comment on that. Interestingly enough, the same holotype that was used in this paper that describes Oriaris is also the same holotype for an animal that was described, Zemidicephalus teasy, that was described 3 or 4 years earlier. Obviously, they've gone ahead and they've published this on the basis of their research and their genetics that they've worked on. So until such time that the ICZN rule on the validity of the earlier publication. The name might be seen as two different names in publication going forward, but again, not for us to comment on the validity of it. We just need to wait on the ICZN to solve and we can sort of move on with our lives going forward with that.
Controversy in the world of reptile taxonomy. Would you believe it? It's not always as clear cut as we would like it to be.
Yeah, I think, mate, as long as the sun comes up tomorrow, there's going to be people arguing about taxonomy. So, nothing changes, nothing's new.
Well, look, either way, it's good to see the genetic work backing it up. It's good to see all the morphology and everything put together in a nice package like that as well. Kind of putting all of the supporting evidence together into a kind of a really convincing bit.
And obviously too, they show that there's some more work to happen as well within this group. So
particularly some of those Vietnamese and northern samples, right? There was some sampling that would really kind of
there was a paper that with some of those Southeast Asian animals that haven't had a name assigned to them. I can't remember what it is off the top of my head, but there was a name that was given to those by some workers out of Europe, I think, from memory. Where they're at with that, I'm not quite sure, but obviously they've regarded them as CF annulatus at the moment. So they're suggesting that they might be different, but they might need a little bit more work to sort out what the hell's going on out there.
Yeah, right. Interesting. And was this the one with the tiger snake reference in it?
I cannot remember.
Let me have a look. I do have to try and find this because I believe there was a tiger snake reference in this paper somewhere in the methods. I'm gonna have to dig through. Bear with me.
Oh yes, I know what you're talking. Oh, was it? No, I thought that was in the next paper.
I'm pretty sure because. Let's have a look. Genetic.
Oh no, here we go. No, it is there on page 143.
143.
Library preparation.
Yeah. I've just got it. Yeah. 143. There it is. Yeah, right. These enzymes. So they're talking about the enzymes that they used for their RADSEC. So RADSEC is a reduced representation library genetic sequencing method, and you use restriction enzymes, in this case PST1 and HPA1, which are added to the samples to break it up, and then you add molecular identifiers and ligators on that, and you kind of sequence it wherever the restriction enzyme cuts up the genome. And they used these two enzymes. These enzymes, just reading from the paper, these enzymes were selected as they have proven informative for population genetics and phylogenetic analyses in the terrestrial elapid tiger snake, Netechus scutatus, in Vicky Thompson. Unpublished data. Unpublished data. There's unpublished tiger snake data. Scott, there's unpublished tiger snake data.
Yeah, it doesn't surprise me, mate.
You're not as excited about unpublished tiger snake genetics as I am.
Oh, I'm always excited about unpublished. Yeah, look, I'm sure there's going to be some interesting things going out there. I suppose the question is whether they're going to sink them or they're going to add to them, I suppose. They do show rapid morphological change in tiger snakes. So, who knows what's going on there, that's for sure.
Yeah, there's a lot of like insular island dwarfism and
and gigantism as well.
Gigantism as well, yeah, absolutely. So very interesting. Vicky Thompson, publish your tiger snake genetics. We're waiting for it. All right, cool. All right, moving on.
Excellent.
What else have we got?
Underwater hearing in sea snakes.
Oh, that's right. Chapuis et al. Oh, yeah, look, there it is. Well, we'll get to that in just a moment. So this is from Underwater Hearing in Sea Snakes Hydrophinae, first evidence of auditory evoked potential thresholds in the Journal of Experimental Biology 2019. This is by Chapuis et al. I hope I'm pronouncing that right. So, reading straight from the abstract here, the viviparous sea snakes are a secondarily aquatic radiation of more than 60 species that possess many phenotypic adaptations to marine life. However, virtually nothing is known of the role and sensitivity of hearing in sea snakes. This study investigated the hearing sensitivity of the fully marine sea snake Hydrophis Stokes Sea by measuring auditory evoked potential audiograms for 2 individuals. AEPs were recorded from 40 hertz, the lowest frequency tested, up to 600 hertz, with a peak in sensitivity identified at 60 hertz. Our data suggests that sea snakes are sensitive to low frequency sounds, but have relatively low sensitivity compared with bony fishes and marine turtles. Additionally, studies are required to understand the role of sound in sea snake life history and further assess these species' vulnerability to anthropogenic noise. Fascinating stuff. So I guess first and foremost, we generally think of snakes as not really having any hearing, right?
Yeah, essentially we think they're deaf. And obviously that's not necessarily the case. I suppose after reading this paper, I read the paper about 4 or 5 months ago, I suppose. And after reading this paper 4 or 5 months ago, I actually changed the way I word my presentations in regards.
And we're back. Minor technical difficulties there. Sorry, everybody. Scott, you were just saying since reading this, you've started changing what you say about snakes in terms of their hearing in some of your presentations.
Yeah. So previously in demonstrations and stuff like that, I'd always referred to them as snakes being deaf. This paper quite clearly shows that they are not deaf. They don't have the same range of hearing that we do, but they definitely do have some auditory perception. And on top of that, there does seem to be some reaction to that auditory perception as well. So I suppose that, I like to make sure that whenever I'm delivering a presentation, I think it's a key thing for anyone delivering presentation is that they are accurate in what they say. And so you can use words to downplay their sensitivity of their hearing, but you don't want to say that something is deaf when quite clearly they may not be.
Right. I mean, we kind of think of hearing in selfish terms, right? Like when it comes to reptiles, the human mammalian inner ear is pretty amazing and complicated. And part of one of the bones of the anvil stirrup mechanism of the inner ear is in reptiles part of the jaw hinge mechanism. It's the quadrate bone, right?
Yep.
So in that, one of the things that allows snakes to open their jaw that wide in mammals has migrated, it's shrunk down and become it's part of the transmission system of the inner ear. So because they're lacking that sophisticated of an inner ear, we go, oh, they must be deaf. But there's always kind of been this idea, and I guess tell me how much credence do you give to this, of like the idea that if a sound wave hit the entire body of a snake, they might be able to pick it up, like using their entire body as kind of a percussive surface, kind of like the tympanium of an ear itself. So it's like they are the eardrum.
Yeah, and I suppose this is where we were looking at the Emetocephalus in the paper before that's got those very small apical pits all over the head of the snake. Certainly in the Pseudoforania polylepis, the Maclay's water snakes that we used to keep here, that they would react from a pressure sensitivity when you vibrate prey within the water near the snake, and that would elicit a strike. And then the snake would eat its fish. So, they certainly do pick up on pressure waves. Whether that pressure wave causes a feed response like in Pseudoforania, or whether it's potentially something that would scare an animal away or something like that, it remains to be seen. You see people have got those vibrators in their their gardens to try and prevent snakes from coming in there. I don't think snakes respond negatively to that sort of vibration. We wouldn't see snakes hit on the roads by trucks and stuff like that if they were scared of that sort of type of vibrational noise. However, in the ocean, as this paper sort of talks about, the drilling processes and those sorts of things might actually be a cause of reduction or displacement in these animals. So, well, you're not.
It's interesting to think of them maybe being affected by seismic activity or seismic testing or or underwater audio testing the same way that maybe some of our larger cetaceans might be.
Yeah,
Whales
definitely.
and dolphins and things like that.
Yeah, definitely. I'm just going to share my screen just so we can see this. Just so we know what we're talking about in this particular animal here.
This is Stokes Sea, right?
This is Hydrophis stokesii doing what Hydrophis stokesii does. This is a fantastic image from Clay Bryce.
Phenomenal.
It's an image that we've used in our book on the snakes of Australia. It's an animal from
Hydrophis stokesii, Stokes Sea Snake for
correct.
The common name.
And so one of the things that these guys mainly eat is they eat these sort of benthic fishes, things like stonefish and frogfish and stuff like that. And that's probably the reasons why they're such a heavily bodied taxon. So this Stokes here has come to the surface and it's eating what appears to be a frogfish. So yeah, pretty awesome image. So now you've got that image of the Stokes Sea Snake. I suppose we can keep talking about this paper, mate, and then I'll stop sharing screen in a second and bring up that figure that we need to talk about.
Before we do can we quickly jump back, because I know you have pictures of the tiger snake and dewgite as well for our audience, just from the last Damon's paper.
Yeah, I can do that. Give me one second.
So the WA tiger snakes, they look a little bit different from I guess the ones that I'm at least more familiar with in Victoria.
Yeah, the WA tigers, they look like they've been hit by every rung on the ugly stick on the way down. I can say that because I keep WA tigers. This animal here is a typical WA tiger.
I would struggle to call that ugly. That's a stunning animal to me.
I suppose it's just a lot of the wild animals have got very high parasite loads and stuff like that.
Right. Yeah. I mean, it obviously looks like it could use an extra feed if it was a captive animal for sure. But I mean, what wild animal doesn't look like that?
Yeah. I mean, that's a captive animal there.
Yeah. A very different look.
And then it goes an all black screen all of a sudden. So
you might want to stop. Oh, there we go.
There we go. Back again. And there it is in its defensive glory there. So basically wanting me to go away. So that's the WA tiger. And we were talking about dugites before as well. Guys, I have to bear with me. I've got about 23,000 images. So, sometimes it can be a little bit difficult finding what I'm after.
I think there's a few herpetological nerds out there who would be happy to just do a podcast cruising through your photo collection. I think that would work just fine.
There's a few interesting things in there. So that's a light one.
Yeah, wow. Look at the head on that thing. That's amazing.
Yeah, so it is a characteristic of a lot of the dugites that they have this spotting here that you can see, which is also gives them the name the spotted brown snake that some people have used. Right. So yeah, they can get quite large and they can be quite defensive like normal brown snakes, I suppose.
All right, cool. Well, look, let's get back to this paper for a moment, if you don't mind.
Yeah. So I've stopped sharing that, so you should be able to go back to.
We'll get back in here because aside from a lot of other things that was great about this paper, I just loved their experimental setup. It is something that is always nice to see some of these zoological experimental setups. And this one was a cracker, I reckon. So
most definitely.
They want to test the AEPs, the auditory evoked potentials of snakes. So you've got them in a sealed chamber inside a wooden box with a speaker attached, and they're anesthetized in a cradle with some electrodes inside them to look at their electrical potentials, evoked electrical potentials in response to, if you can see, this big bass speaker down here at the bottom, and it's sitting on a nice big bit of rubber to minimize even the ground vibration kind of going up into there. So yeah, that's that's their amazing experimental setup. I love that diagram. It's fantastic.
I especially love the car tire that they use to sit it on. You spend all get it all right, and they go, right, we'll sit it on a car tire to stop vibration. But at the same time, the simplicity of it is great too.
Yeah, and look, that's sometimes the best. Simplicity is a lot of the time the way to go. Why overcomplicate something? But yeah, snake in a box with a speaker and some cables. Yeah, very cool. So let's just scroll down here very quickly to the end of their discussion. In this study, we showed that the sea snake Hydrophistocus can detect underwater sounds of low frequency with a relatively low sensitivity compared to other aquatic vertebrates. So still relatively low compared to, let's say, fish or, even I guess some of the other
cetaceans,
mammals, and things like that. Yeah. To further assess the acoustic ecology of sea snakes, more studies are required to discriminate whether sea snakes are primarily responsive to sound pressure or particle motion. As sea snakes are ecologically very diverse, it will be critical to explore the auditory sensitivity of other sea snake species and to examine the hearing abilities of sea snakes at different life stages. To assess the potential influence of anthropogenic noise on sea snake persistence, further work should focus on assessing the individual and population level responses of these animals to noise and the effect of noise on critical behaviors, mating, feeding, and anti-predatory behavior, physiology such as auditory threshold shift, and anatomy such as scale mechanoreceptors, inner ear, and lung damage. The idea of lung damage from like percussive noise, that's just insane to think about. But I guess it must happen on a fairly regular basis. Who knows?
Yeah, it's quite scary. There's a place off the northwest coast of Western Australia called Ashmore Reef, and it had, not only did it have some of the highest diversities in diversity of species in Western Australia, but it also had quite massive numbers as well. I've spoken to Hal Cogger about some of his initial collecting in the 60s out on Ashmore, and the sheer numbers of sea snakes over there. And you basically go there at low tide and you flip over pieces of coral, and you'd find things like Apiciurus, Apifrontalis, and Foliosquama and stuff like that. You go there, they started seeing much reduced numbers in the 80s and 90s, and now subsequent investigations over there have not detected some of these taxons in about 20 years. Now, they're not sure why those sea snakes aren't there anymore, but the suggestion of noise and fishing with dynamite and all that sort of stuff is potentially one of the issues. I mean, Ashmore Reef is right on the edge of the Australian territory, and so potentially some of the impacts in international waters and Indonesia, Indonesian fishing vessels and stuff like that could be causing issues for our wildlife, I suppose. And this is just maybe potentially another piece in the puzzle.
Yeah, it was definitely interesting to, I think one of the points that they made there at the end was as well the trophic and I guess ecological diversity of sea snakes. there's probably going to be some that would not be massively affected by anthropogenic noise, but considering their diversity, like it's almost certain that there would be some that would. Like if there is a range of sensitivity in these animals, then with the amount of diversity that they show, it seems like something is going to stick. They have quite a wide range of prey and quite a wide range of habitats that they use, whether it's the seagrass meadows or the coral reefs or the open ocean ones like the pelagic yellow-bellied sea snake. They're kind of such a diverse group, and they have so many different types of life histories that it's quite likely that some of them are going to be at risk.
Well, I mean, the other thing too to remember is, it's, we think of vision being a primary sense, and we think of vision being one of the more significant senses for snakes, obviously, along with sense of taste or smell. In an aquatic habitat, particularly something that's got very turbid water, such as an estuary or something like that, vision's not going to be very effective. So, if you remove one of those senses essentially from something, then it's well known that you see the other senses increase to compensate for it, so to speak. So potentially that could be another issue that we're seeing here, that that the sea snakes have actually got a elevated level of sound sensitivity compared to other elapids, and that to me would actually be a really interesting point to check to see. I mean, the aquatic sea snakes are essentially aquatic elapids. It would be very interesting to test some of the Australian elapids for their sensitivity to auditory stimulation, so to speak. Maybe there is something to it, and maybe that you do need to be quiet when you go hunting tiger snakes or big ice or something like that. Maybe that was the factor why they can not necessarily find them every time. Maybe there's noise that plays a part, and we're just not picking up on it at the moment, is we're assuming that they can't hear us.
Yeah, quite possibly. And, maybe we're just coming across the more bold ones, the ones that don't run away, but they still do hear us, right?
Definitely.
Yeah. All right, let's move on here onto our next. And obviously we're going through quite a few here, so we're only going to touch on some of these, such as this one from Zoo Systematics and Evolution, a new species of green pit viper of the genus Trimesurus lacipeda 1804 Reptilia serpentis viperidae from western Arunachal Pradesh, India. This is by Mirza et al. Largely, we picked this animal because it's got a cool name. A new species of green pit viper of the genus Trimesurus is described from the lowlands of western Arunachal Pradesh state of India. The new species Trimesurus salazar is a member of the genus Trimesurus, relationship deduced contingent on two mitochondrial genes, 16S and ND4, and recovered as sister to Trimesurus septentrialis. Septentrialis. The new species differs from the latter in bearing an orange to reddish stripe running from the lower border of the eye to the posterior part of the head in males, high number of pterygoid and dentary teeth, and a short bilobed hemipenes. Description of the new species and T. aruncalensis from northeastern India in a span of less than 1 year highlights the need for dedicated surveys to document biodiversity across northeastern India. So Trimesurus salazar, for the nerdier people out there, this is actually named after Salazar Slytherin, the one of the co-founders of Hogwarts School of Witchcraft and Wizardry, and the namesake of the Slytherin School of Witchcraft. And let's have a look at this thing. It is quite a beautiful, like all the Trimesurus, fairly beautiful animals, right?
Yeah, they're pretty snakes. It's a pretty horrible name, but
yeah, you're not down for the Slytherin name?
Oh, look, there's a whole heap of other names you probably could have given it, but whatever. At the end of the day, maybe I suppose there is one thing about it, is that it's been picked up by a lot of the news outlets, and it's been picked up by a lot of places simply because it's named after a entity within Harry Potter, I suppose. Interesting etymology that you've got essentially a patronym after a non-existent being. It's a very interesting thing. So,
well, yeah. Look, let's actually read the etymology because they have it here somewhere. That's actually a great shot of it, though. Look at that, pretty cool.
Yeah, that's a juvenile animal there from 2008.
Yeah,
you'll see how it's got that red tail. That red tail is probably indicative of quarter luring when they're younger.
Right. So like our death adders, it probably uses that to wiggle around near its head and have.
So they'll wiggle that around and that. Yeah, they do wiggle that around, and that is potentially to help them get geckos and small lizards.
Very cool. Very cool. Yeah, interesting. Let's see if we can find this etymology here because it was.
It's
the specific epithet is a noun in apposition for J.K. Rowling's fictional Hogwarts School of Witchcraft and Wizardry's co-founder Salazar Slytherin. He was a Parselmouth that links him to serpents. Suggested common name Salazar's pit viper. So there we go. Salazar's pit viper. They've named a snake after a Harry Potter. Founding Hogwarts character, which I think is pretty cool. Interesting.
I suppose what I was I think the reason that they named it this, aside from whether or not it's a cool thing or not, I don't like the idea of it myself. But I didn't name it, so hey, more power. The thing I think that one of the benefits is that it has been picked up by these news outlets. And so by being picked up by these news outlets, it gets people interested in the work that these guys have done collectively as a group, and potentially might help them with funding some of their other work that is out there. I mean, they described a lot of the same crew described that that other Tremoceurus from there. And it hasn't had anywhere near the notoriety that this thing has had. So the fact that thing was brown and was spotty and wasn't bright emerald green like this thing is, and it doesn't have a
cool name,
and didn't get this name that's nice and easy to pronounce and all the rest of it, then maybe that's why it was it's been picked up the way it has. So.
Look, I guess in the end, anything that kind of helps biodiversity sciences get a little bit more shine, a bit more attention, not necessarily a bad thing. I guess while we are here looking at the phylogeny here for some of the genetics nodes, you might see some pretty lowly supported nodes here at the base of the Trimecerus salazar tree, where septentrionalis and fasciolatus and insularus break off. There's actually some pretty low support values. I was concerned about that as well. But this is all couched in Malorda and Thorpe's Phylogenies. Here you can see in the discussion in 2000 and 2004, which are very, very complete for the Trimecerus, actually have really, really good support values. And while this tree doesn't have the great support values, it shows similar topologies. So it's not like branching away in different directions from what the more supported stuff by Thorpe and co are showing, which is nice.
Did they only use mitochondrial DNA in this?
I do not recall. I think it was ND4. I think they probably used 2 mitochondrial genes. Let me scroll on up. Molecular analysis, genomic DNA isolated, 16S RNA, mitochondrial, subunit 4, ND4 and 16S. So yeah, 2 mitochondrial genes.
Yeah. I must admit, the other thing that was really cool about this paper was the micro CT scans of the skull.
Fantastic, right?
Nuts.
There you go.
Yeah.
Look at those. Yeah, great to see, right?
Yeah, most definitely.
Beautiful. All right, and we have one more to touch on. I know it's been a long one. I know we're both tired,
especially after our technical issues today.
Oh yeah, so many. All right, and moving on. on to this latest one, which is a very recent one, 2020, when Piaget additions to the phylogeny of colubrine snakes in southwestern Asia with description of a new genus and species. This is by Raja Bizadeh. I'm totally butchering that, just like I always do. Reptiles are still being described worldwide at a pace of hundreds of species a year, while many discoveries are from remote tropical areas. Biodiverse arid regions still harbor many novel taxa. Here we present an updated phylogeny of colubrid snakes from the Western Palearctic by reanalyzing a super matrix of all available global snake species with molecular data and report on the discovery of a new genus and species of colubrine snakes from southeastern Iran. The new taxon named Persiophis fahimi, genre nov, species nov, is nested within a clade containing Middle Eastern and South Asian ground races such as Litorhynchus, Rhynchocochlomus, Wallaceophis, and Wallophis. This species has a derived morphology including an edentulous pterygoid. What does edentulous mean? Edentulous, so missing teeth. Edentulous pterygoid, so no teeth on the pterygoid bone, and occurrence of short and blunt teeth on the palatine, maxillae, and dentary bones, an elongated snout, and a relatively trihedral first supralabial scale that is slightly bigger than the second and elongated towards the tip of rostral. We also report on the osteology and phylogenetic placement of several poorly studied colubrines, Hierophis andrianus, reassigned to Dolichophis, and Mutarophis baroni. So those last two species, they kind of reassign Hierophis andrianus to a different genus. But the most interesting part here is definitely Persiophis fahimi, which I believe they discovered in
It's Fahimi.
Fahimi, of course.
Fahimi, because it's a double I, you've got the EI.
Yes. EI. Yes, indeed. I believe they picked up the first specimens in like 2006, 2008. Yeah, there we go. A specimen of color superficially resembling snakes of the genus of the genre Rhynchocochlomus or Litorhynchus. Voucher specimens were preserved and deposited at the International Center for Science, High Technology in Iran, and tissue samples were collected for molecular phylogenetic analysis. So these they kind of jammed into for a lot of snake nerds. You guys will know Pyron's paper or Figueroa's paper here as well. Pyron did the supermatrix of pretty much all extant squamates, right? So they had this big, massive mitochondrial, massive genetic tree of a lot of genes of all of the squamates. Figueroa here is, I believe, a similar approach, and I believe worked along with the side with Pyron, who did the supermatrix of squamates, and this one was specifically for serpentates, so for snakes only. And they've used that supermatrix, which you can kind of see here in this paper. They also did some very cool osteology, by the way, in this paper. Very cool. But here on the left, that is the giant supermatrix of snakes, right? This whole thing here, which you can't even see the taxon anymore because of, all right, look at that thing. It's massive. And then you take this tiny segment of it and you kind of get these West Palearctic Asian colubrids right here. So that little highlighted segment that you can kind of see at the middle of the page, that's what we're seeing there in B. And
I think what needs to be just noted with Pyron's stuff and this supermatrix tree is that not every taxa, not every species has been put into this. It's only the ones that they've had samples for.
Absolutely.
So there is, when you look at the Australasian or lapid radiation that sits within this paper, there's probably about 70 odd species that haven't been included into it. So, you know, you've got much lower values. I mean, that you don't have
the
diversity.
Support values for a lot of nodes
it's fantastic. There's a lot there, but
taxa in between,
but there's a lot of missing taxa in there as well. And then not only does that need to be taken into consideration, but on top of that as well is that how many animals have been sequenced in that? Is it Are we talking 1, 2, 3, 4, 5 animals from a particular taxa, or are we only looking at the, if we look at this particular tree, what they're looking at the moment, towards the top, you've got 3 instances of Litorhynchus diadema in there. So you've got, is that, did they only look at 3? And you've got quite deep nodes there within that group that's potentially showing that there could be some variation within diadema as well.
Well, yeah, there definitely is, but I know for a fact that they only had 1 Perseophis fahemii because that's all they found. That's all that's ever been found.
Yep. Yeah, it's described. It's only described from a single taxon.
Yeah, and they've been back looking for it multiple times, and they have not found it. So while and you can see from the branching, it does split off very early. It is very divergent from the rest of them, but there could be plenty of intervening genre in there that is still yet to be discovered as well. This could be very, very incomplete. That being said, very, very divergent. How much overlap there is in any of those genetic characters, we might not know until we get a fully complete tree. Once we find some more species, who knows how many more there is out there to find, right?
Oh, most definitely. And I suppose the point that I was trying to make is that, and not very effectively, is that, in the case of, there's plenty of taxa out there that are currently sitting lumped within a single taxon. And, a lot of the blind snakes, that's a great example of that. Something like Anilios bituberculatus or Anilios guntheri or Grypus are species that are most likely to be species complexes. And so if you placed those into the tree and you're only looking at one example of Grypus or bituberculatus or guntheri, then where they sit within the tree is fine for the animal that's been sampled, but that may not be the case for all of the guntheris or all of the Grypus or all of the bituberculatus.
Right. Well, yeah, it is always hard to capture that kind of amount of diversity for phylogenetics from just one sample, but sometimes that's all you got to work with. Good times.
At the end of the day, I mean, it's a pretty incredible sort of West Iranian colubrid. That the one thing that was of note there to me was that I think there's a photo of it. I'm pulling it up.
There we go.
That supralabial scale, that first supralabial. That's a really unusual feature to have that enlarged supralabial, first supralabial. So you'll see there that
that one right, like towards the tip of the snout?
Yeah, so usually this first supralabial is smaller than the second and third.
Yeah.
And so to have that that supralabial there being so long is quite unusual.
Yeah, right.
Very pointed note.
It does have a very,
I wonder if these things go into crevices.
Yeah, very elongate, maybe like even dorsolaterally compressed sort of head, but the body doesn't seem to be quite so flattened.
So it looks like a big-eyed, narrow-snouted thing. So, but it doesn't seem to have really sharp teeth. So
who knows what it's eating? That's the weird part. No teeth on the pterygoid bone, but and small blunt little teeth, right?
So the one thing that the one group of snakes that don't have teeth essentially is Dasypeltis, the egg-eating snake. So this having that elongated snout and elongated body, can you just go down to that CT scan again?
Yeah. There we go.
It'd be really interesting to see a CT scan of the whole body and see if there's any projections on the base of the ribs, because this may be an animal that tends to eat reptile eggs as opposed to, I don't think it's eating bird eggs, but it might be a reptile egg eater at a guess. It's got some very weird sort of skull morphology, that's for sure.
Yeah. Can you see figure D there at the bottom with the sagittal view of the skull?
Yeah.
Kind of see some, are those ribs down there along the edge? Or is
They just look like the articulations off the bottom of the
rib? Off the vertebrae.
So when you look at a Dasypeltis, they've actually got projections off the bottom of the spinal column.
Right, that help them kind of like crank their neck once they've got the egg in their throat.
Yeah, and they then rupture the egg. That's what cracks the egg. So that might. But some of the other egg-eating colubrids have got long teeth that sort of slash the egg open, and that's how they get it down. So who knows? Maybe it's eating insect eggs or something like that. It's a very
well. Look, there's we've already had a look at a fish egg eating sea snake tonight. So,
well, I suppose the point that I make is that if you go up a little bit, you've got that dentition shot, or the lack thereof, I suppose. It would not look like that would be a very effective grab for that snake to hold onto a lizard in that jaw.
No, definitely not. Do we have another good CT somewhere with these guys had the Cephalus? Did they do a CT here as well for us to look at?
I don't think there was I don't think they had one of it.
Oh, okay. This one did. Here we go. So something with actual teeth. The Trimesurus. So let's have a look at the CT.
Yeah, so that Trimesurus, you can see that how elongated those teeth are for helping hold their prey. Obviously, they've got those two big fangs that can articulate. Interesting that animal's actually got two fangs on the right side there as well.
He's got a double set.
Well, it's they obviously as they because those fangs are so important. As one drops away, you've got another one there that's ready.
But yeah, compare that to the level of dentition here on Mr. Gummy.
Yeah.
Plesiophus.
Yeah, it would have been nice. They could have called it the Gummy Snake. That would have been a good.
You don't have any other Harry Potter references you want to throw at this one?
No, mate, no. Not at all. Very, very cool snake though.
Yeah, fantastic. All right. Well, look, I think that's pretty much all we have for now. Did you have any other notes that you wanted to chat about, or that's pretty much it for our first set of these Quarantinis?
No, mate, that's it. I think we got there in the end. This is our second crack at this. So,
second crack at an actual podcast, and maybe 20, 30 test recordings to try to get this this whole circus, this whole goat rodeo, functional.
Well, we had a bit of fun with it anyway, so that's not the end of the world.
Yeah, well, no, I'm glad you had fun, man. I certainly did. I'm glad we had to got to talk a bunch of topics today. We actually covered quite a lot. So, man, I had a great time. I hope you did as well. Everybody, you can check out Nature for You at wildlifedemonstrations.com or on Facebook at Wildlife Demonstrations. Where else can people catch you?
Look, honestly, they can jump. Easiest way to get me at the moment is through the website. We've got 3 books there at the moment that we're happy to move on. We can move them on to those ones. So yeah, get onto it. Get the books. Yep, that's all of them. So yeah, I mean, look, we're working on other ones as well, but yeah, look, get on it. Buy the books to help support the authors is always nice.
All right, man. Well, look, thanks again, man. It's been a pleasure talking to you as always. I hope you and Ty are surviving the quarantine well. I'm sure once it's over, we'll actually have a proper catch up at some point. But appreciate your time anyway, and much love to everybody over there on the south side.
Happy days, mate. Enjoy yourself up there.
All right, man. Will do. Thank you very much, man.
All right. See ya. Bye.